Hello guys, welcome back to my EduSpace. Hope you all are doing and learning well. So in the last video, we have discussed about the structure of cell membrane. What are the constituents involved in the formation of cell membrane? If you haven't watched out yet, go and watch it out. I will provide link somewhere in this video. So in continuation with the plasma membrane, let's discuss about the membrane proteins, that is integral as well as peripheral proteins. So without taking much time, let's get started. Introduction. As we all know that cell membrane consists of phospholipids and proteins. Okay, they both constitute the major part of cell membrane. Beside this, cholesterol and oligosaccharide molecules are also present. Basically, phospholipids are responsible for establishing semi-permeability to the cell membrane. The proteins, they act like mothers in the houses. As mother performs all the functions of a house, just like that in cell membrane, proteins act like mothers and are responsible to carry out all the functions of cell membrane. Now, the constituent ratio varies. It means lipid and protein ratio in cell membrane varies. It depends on the type of cell. So it is not in constant ratio, but they varies, which depends upon the type of cell. Like for instance, in mitochondrial membrane, it contains high percentage of proteins because it requires more transportation for production of more energy. So here the protein percentage is much higher as compared to the lipid percentage. In myelinated neurons, there is high percentage of phospholipids than proteins because phospholipid provides insulation to myelinated neurons. Generally, in most of the cell, the ratio is just half-half. 50% cell membrane consists of phospholipids and 50% of proteins. So next is types of membrane proteins. Basically, membrane proteins are of two types. The integral proteins, or we can say intrinsic proteins, because they are permanent part of membrane and associated strongly with hydrophobic tail of lipid bilayer. And the next is peripheral protein, or also known as extrinsic proteins. These are transiently or loosely associated either with integral proteins or with lipid bilayer of cell membrane. These associations are weaker as compared to integral proteins. So let's read the slide. Integral and peripheral proteins are the two types of membrane proteins in the phospholipid bilayer. Integral proteins penetrate the hydrophobic core of the lipid bilayer, while peripheral proteins are attached to the intracellular or extracellular surface of the lipid bilayer. The membrane proteins are free to move within the lipid bilayer as a result of its fluidity. So next, integral proteins. Let's discuss this with the help of the diagram. So you can see over here, these blue colored, this, which are embedded structures are integral proteins. Okay, these are integral proteins. They look like completely immersed in the lipid bilayer. See, these are looks like that they are immersed in this lipid bilayers. Okay, they are having both hydrophobic as well as hydrophilic regions. See, the area that spans the hydrophobic core of lipid bilayer is also hydrophobic regions of a protein, and the area this and this which faces at the cytosolic and or at the extracellular ends is hydrophilic regions or polar region of proteins, because it is made up of polar amino acids. That's why these integral proteins are also known as amphithetic. Okay. The middle part of these proteins that are immersed inside the hydrophobic core of lipid bilayer is composed of, this one is composed of non-polar amino acids. Okay, non-polar amino acids. Therefore, this portion is associated strongly with non-polar fatty acid tails of lipid bilayer. Okay. Besides this, they are also having hydrophilic region, this, on both sides of the lipid bilayer, here and here, that associated with the polar head of lipid bilayer. So we can say that these integral proteins are strongly associated with the lipid bilayer. Okay. So integral proteins are embedded within the lipid bilayer. These bind to the membrane extensively and span the entire width of the lipid bilayer. Therefore, they are known as transmembrane proteins. Why these are known as transmembrane proteins? Because they span the entire width of cell membrane. Next. So let's take another picture to get understand well. So these all four are integral protein types. Some span the entire lipid membrane, okay, at once, and some forming helices and span multiple times like this. We're seeing this particular protein spanning the lipid bilayer multiple times, and this one spanning only at once, and some forming channels like this protein forming channels, and this type of proteins are also known as channel proteins, which are having the hydrophobic tunnel. This is having hydrophobic tunnel, and some are attached to only one lipid layer of cell membrane like this. This particular protein is attached to either inner membrane of cell or either at outer membrane of plasma membrane. So this type of proteins which attached to only one leaflet of cell membrane is known as monotopic integral proteins. So the type of protein that are attached to only one single lipid layer, either with the inner membrane or sometimes at the outer membrane, and they do not span the entire lipid bilayer. Okay, these are known as monotopic integral proteins. The examples of this type of monotopic integral proteins are prostaglandin S2 synthase. Examples is prostaglandin gliadin H2 synthase. Basically, it is an enzyme that help in the formation of prostaglandin H2. Next. So I hope you understood why these integral proteins are known as transmembrane proteins, because they span or cross the entire lipid bilayer. Now, on the basis of how many times these proteins span or cross the lipid bilayer, they are classified into two types. The first one is single pass protein, or also known as biotopic proteins, as they span the entire lipid bilayer only at one time. And the second one is multi-pass proteins or polytopic, as they span the lipid bilayer more than once. You can see over here, these particular proteins are spanning multiple times in the lipid bilayer. Okay. Now next. Now the alpha helical and beta barrel configuration. As we know that alpha helix and beta pleated sheets are secondary structures of protein. The integral proteins that adopts an alpha helical configuration, this is in this form. If it occupies this alpha helical arrangement, then it is known as alpha helical configuration. During spanning the lipid bilayer, basically all types of biological membranes are having alpha helical configurations. The example includes Bacteriorhodopsin. It's present in Archaebacteria. It uses light to establish proton gradient. It occupies mostly alpha helices that contain non-polar amino acids. Basically, this particular reason, most of the helices is composed of non-polar amino acids that interact strongly with the non-polar or hydrophobic core of lipid bilayer. Next, the integral proteins can have beta barrel configuration where protein occupies beta pleated shapes. It is exclusively present in chloroplast, mitochondria, and outer membrane of gram-negative bacteria. Example of beta barrel proteins is porins. It acts as a channel. Basically, porins are example of channel protein. So the porins acts as a channel. It consists of beta sheets. You can see over here, these are the sheets. Okay. This is the secondary structure of protein. Beta sheets having alternate non-polar and polar amino acids. The non-polar amino acids interact with non-polar tail of phospholipids, and polar amino acids forms the interior of the channel. So this beta pleated sheets are having non-polar as well as polar protein amino acids, as well as polar amino acids. The polar amino acids and non-polar amino acids are alternatively arranged. The non-polar amino acids interact with the non-polar tail of phospholipids, and polar amino acids forms the interior of the channel, means the tunnel of the channel is completely made up of polar amino acids. So it allows the polar molecules to come across the cell membrane without any difficulty. Next. So, transmembrane proteins are the only class of proteins that can perform functions both inside as well as outside, because they span the entire lipid layer, which is present. Some part is present at the extracellular space, okay, and some part is present at the cytosolic space. So that's why these are known as transmembrane proteins, and they act functionally inside the cell as well as outside the cell. The transmembrane proteins have hydrophobic part that are associated with hydrophobic core of lipid bilayer. As a result of these, transmembrane proteins get stuck permanently in cell membrane and are very hard to isolate. So they are amphithetic in nature. They are having both hydrophobic part as well as hydrophilic part. So they get stuck permanently in the cell membrane and are very hard to isolate them. They can remove from the cell membrane with the use of harsh detergents that destroy the lipid bilayer. Once the lipid bilayer is destroyed, then they can only get isolated. So, peripheral proteins. After integral proteins that were extensively associated with lipid layer, there are some other types of proteins whose associations are very weak. So you can see over here in this diagram, this is the cytosolic phase and this is the extracellular matrix. Okay, there are some proteins, they are known as integral proteins, and you can see in the green, these are known as peripheral proteins. Okay, these are known as peripheral proteins. They can attach with the lipid bilayer, like in this protein is peripheral, which attached to the lipid bilayer. Okay, and these two proteins are attached to the integral proteins. They bind less extensively with lipid bilayer than that of integral proteins. They bind via ionic bonds or hydrogen bonds with the polar heads of phospholipids or onto the polar region of integral proteins. As I just discussed, they can bind to the lipid bilayer or with the integral proteins, may be present at the cytosolic end or at the extracellular matrix, because their interactions are having ionic bonds or hydrogen bonds, which are considered as weaker interactions. They can easily separate from the lipid bilayer and able to removed without harming the lipid bilayer. Okay, peripheral proteins are less mobile within the lipid bilayer. Next, yeah. So there are some proteins that associated with cell membrane having only interactions with lipid tail, either of inner membrane or of outer membrane of lipid bilayer. So you can see in this picture, there are two lipid anchored proteins. The first one is this and the second one is this. These are two lipid anchored proteins because they are anchored to the lipid tail of cell membrane. This one which anchored with the lipid tail of inner membrane. Therefore, it is on the cytosolic side, whereas this protein is anchored at the lipid tail of outer membrane. This one is outer membrane and this one is inner membrane. So this is present at the extracellular space and this one is present at the cytosolic end of a cell. So they attach to only a single layer of phospholipids out of the two layers and mostly anchored at the lipid tail of phospholipid layer. Now the functions of membrane proteins. They help in cell adhesion, they help in signal transduction, they act as receptors, they act as enzymes, and they help in transporting molecules and ions throughout the lipid bilayer. Thanks for watching.